On behalf of Cambridge Health Tech Institute's Global Web Symposia and our sponsor for today's event, Agena Bioscience, I'd like to welcome you all to Liquid Biopsy, applications of circulating tumor cells and circulating tumor DNA as a non-invasive tumor tracker. I'm Allison Proffitt, editor of BioIT World, and I'm the moderator for today's event. Now I'd like to introduce our two presenters for today. Dr. Pierre-Jean Lamy is an oncogeneticist specializing in biomarkers of solid tumors. Dr. Lamy is head of Imagenome at the Medical Institute for Genomic Analysis, which is part of the first private clinical laboratory group in France. Imagenome provides molecular tests that help clinicians with cancer diagnostics and personalized cancer therapy. Darryl Irwin is the Senior Director of Applications Development at Agena Bioscience. Dr. Irwin manages scientific operations in the Asia-Pacific region, including customer support, genetic services, marketing, and product development activities. Welcome to you both. And Dr. Lamy, would you get us started?
Thank you, Alison. Hi, everybody. I'm Dr. Lamy from Medical Institute of Genomic Analysis in Montpellier, France, and I will share with you some information about the use of circulating tumor cells and ctDNA as a non-invasive tumor marker. So we are at a very exciting time, the era of personalized medicine, an innovative approach to treat a patient, taking into account individual differences in patient for cancer, individual differences in tumor-specific genetic alteration. And for that, it's necessary to get the good techniques, but also the good samples to give the right treatment to the right person. So what is liquid biopsy? Liquid biopsy is the other way to get information about without invasive biopsy. It is related with the fact that tumor content could be released into the blood circulation. And as you see in this slide, we can get some proteins, as well-known tumor markers, but also circulating tumor cells, and different kinds of DNA, like cell-free DNA, viral DNA, methylated DNA, and also microsatellites and nucleosomes. So let's have a look on, first of all, circulating tumor cells. Circulating tumor cells and also disseminated tumor cells can be found in the bone marrow and also in the bloodstream, and they are related with the process of invasion and metastasis. We can find them in different stages of the disease, from early detection to the detection of the relapse, but of course, due to a lack of sensitivity, it can be used essentially in patients with metastatic disease. So how to catch CTCs? It's not very easy. It's like to find a needle in a haystack. Normally, there is no circulating epithelial cells in blood. And even in advanced cancer, CTCs are very rare. So we can catch them with different techniques using physical property of those cells. Those cells are a little bit higher than normal cells. Bigger than normal cells, and so filtration, but also density could be used, but this is not specific methods. And also we can use biological properties using, for example, iron particles that are coated with specific antibody against specific antigens from those epithelial cells. And usually this antigen is EpCAM. There is also negative selection with CD45 antibodies to discard white blood cells. The only system that is FDA-cleared is the cell search system that can be used for diagnostic, for example. And this system is based on a positive selection with EpCAM antibodies on iron particles. The cells from one tube of blood are trapped by the system, catched by the system, and then they are spot system to allow an image analyzing. And as you see, there are different channels to detect some specific antigen the cells, like cytokeratin, that before variability. You have also a channel for a negative selection with CD45, antibodies, and one channel that is open for different analysis. Here in this slide, you can see the HER2 protein that is targeted by antibodies. So this system leads to many publications. And if we look at the clinical application of the CTCs, first of all, they were focused on the enumeration of cells, and the enumeration, the quantification of the number of cells is related with prognostic, and also it can be used for the detection of recurrence in breast, prostate, non-small cell lung cancer, also in colorectal cancer. And this is just here the Kaplan-Meier curve according the survival, disease-free survival of patients with non-small cell lung cancer according the number of cells below or upper the threshold that is 5 CTCs. But we can also now catch those cells and analyze those cells to determine the molecular profile of those cells. And there are more and more information about that leading to the determination of the prognostic, but also the predictive value in response to therapy. And a great example of that is the detection of RNA specific splicing variant B7 of androgen receptor in prostate cancer. And this variant is related with a non-functional protein, non-functional androgen receptor correlated with relapse of patients treated with anti-hormonal therapy. In the future, we can expect to get more of those cells because, as I said before, this is rare event, so it's difficult to catch them, it's difficult to analyze them, so samples does matter, and there is some system now to analyze those cells or to catch those cells directly from the vein of the patient, and in the lab on a shift, those cells could be trapped, analyzed, and then you can do qPCR, staining, enumeration, also next generation sequencing or FISH analysis, what you want. So molecular CTCs characterization is very important. It could be a very important development in the future. Let's have a look now on circulating cell-free tumor DNA. So, as you know, there is two main fields in medicine that use now circulating cell-free DNA. Cell-free DNA coming from fetus, the fetal DNA from non-invasive prenatal diagnostic. Is this my father? Is this a boy? Is this a girl? Does my baby have this Down syndrome? So this is a very important field. It was developed by Dennis Slow. And also the analysis of tumor in cancer and in autoimmunity, transplant, and also infection. And as you see here in this slide, you can use blood, but also urine, or different fluids, like also CSF, for example, in these different conditions. So the story of ctDNA is a long story. It was a quiet story, but a long story. It begins in 1948, and I'm very proud to see you this slide of a French publication by Mandel et Mette in a very well unknown publication, Compte rendu de biologie de Paris. And those researchers showed that they can quantify nucleic acid, RNA, and also DNA in different conditions like sepsis, heart disease, tuberculosis, and even pregnancy. But we had to wait like the beginning of the 21st century to show an increased number of publications and the increased interest, particularly in the field of oncology. And as you see, those publications in the 17th, the 18th were not read by a lot of people, but they exist. And now, according to the huge development of molecular biology in oncology, of course, it was a tremendous change in the interest of this analyte. So there are many debates on the size of circulating DNA, many debates. And as you see here, we can now have an accordance that the peak of ctDNA is around 150 base pair. It is related with the apoptosis. Apoptosis lead to oligonucleosomal fragments that have this size of 150, 166 base pair. And most of the circulating DNA we can catch is produced by apoptosis. Necrosis that can also appear in cancer leads to longer fragments, around 300 base pair. But if we look at circulating DNA, there is some confusing data because there is not only circulating tumor DNA, but also non-neoplastic DNA. And in this publication by Penn, you can see by atomic force microscopy that DNA from healthy donor is a little bit longer than cell-free DNA from cancer patient. And it is very important now if we want to look at specific cell-free tumor DNA to look at this small fragment. So cell-free tumor DNA is present in various cancers, in almost all cancer, but at a different level. As you can see here, in bladder, colorectal, ovarian, breast cancer, the level is quite high. But in cancer like prostate, it's a little bit lower, and in thyroid and glioma, maybe due to the blood-brain barrier, the quantity of cell-free DNA is very low. Circulating tumor DNA is related with tumor burden. In this publication made by our team in Montpellier, it was a publication made with a mouse in which we xenographed some cell line, SW620. Those cell lines present a mutation in the gene KRAS, and you see there is no relation between the cell-free DNA from the mouse according to the tumor weight. But on the right side, you can see there is a good correlation between the tumor weight and the quantity of specific human KRAS cell-free DNA, KRAS mutated cell-free DNA. There is also a relation between cell-free DNA quantity and survival. And there are many publications on this topic, just this one to show you that in different patients with different metastatic origin, there is an inverse correlation between the quantity of cell-free DNA and the survival. So if we want to analyze this cell-free DNA, it's very important to know some data on pre-analytical issues we know very well now. So it's necessary to use plasma better than serum because during the coagulation, white blood cells could release non-specific DNA and it increases the background. EDTA for plasma and no heparin because, of course, usually after the extraction, you have to use qPCR and heparin could inhibit qPCR. And then you have to get high volume. I said at 3 milliliters at least, because it could improve the sensitivity. And sensitivity is very important, we'll see later. Because of the quick release of normal DNA by white blood cells, it's also necessary sometimes to use stabilization tubes like STREC tubes or Roche DNA or PAXgene tubes. The double centrifugation is necessary with high speed, at least one at high speed centrifugation in order to discard exosome or other vesicles that can also absorb long strain of DNA, non-specific DNA. Extraction is usually used with specific kits that work very well, like this example from Roche of Qiagen, and there is other. And the quantification, you have to remind that you have to quantitate small fragments, so qPCR is better than spectrophotometry. And now we have to move to the challenge of the cell-free DNA detection. Of course, we can use different techniques. If we have a lot of cell-free DNA, this is not a problem. Even single sequencing could work. But usually, at the very early stage of the disease, or in order to monitor patient, you can get only a few percentage of cell-free tumor DNA. So you have to use very specific methods like allele-specific PCR, beaming, digital PCR, on MassARRAY with a new system, very sensitive called UltraSeq, that will show you this test in the next presentation. So what can we analyze in cell-free DNA? Exactly what we can analyze in tumor DNA. Quantification, qualification, detection of molecular aberration, viral DNA, quantification also of mitochondrial DNA could be important. And also, we have no time to develop now, but analysis also on cell-free RNA. So there are many publications that showed we can use cell-free DNA to detect actionable mutation. And this is just only one example by Janko in Oncology last 2 years, showing the very good concordance between archival tissue and plasma cell-free DNA. And as you see, these results are very good. We, in a clinical trial, prospective clinical trial, double-blind study, we show exactly the same results with a very good accuracy between the tumor test tissue and tumor tissue test and the cell-free DNA analysis. So the question is now, can we monitor patient during the pathology, during the follow-up? And here in colorectal cancer, this is only 2 examples of patient 1 and patient 2. On patient 1, you can see the decrease of cell-free DNA after resection of the tumor, then a long phase of no elevation and no clinical symptoms, and then an elevation before the positivity by the CT scan. The relapse, the local tumor was destroyed by surgery, and then you can see an elevation again of specific biomarkers. It is exactly the same with the CA, that is a very good tumor marker in colorectal cancer. For patient 2, exactly at the beginning, the same result, a drop of the quantity of cell-free DNA, exactly like a CEA, and then no relapse. The CT scan was completely white, okay, and again, during the follow-up for this patient with no metastasis. Another example in breast cancer, because in breast cancer, you cannot look at just one mutation. There is no public mutation in this cancer. And in this trial, they get just one sample from a breast cancer with a biopsy, and next perform NGS analysis to get different information about the mutant content, tumor content. And after neoadjuvant chemotherapy and surgery, they get some samples, blood samples, every 6 months. And I will show you only one patient, but that is very relevant. As you see, all the biomarkers on cell-free DNA dropped after chemo and surgery and stay at a very low level with no problem. But only one, the red one, stay at a very high level and then raised. And this elevation was before the CT scan. The first CT scan at 12 months was negative, but it was positive at 18 months. So you can see the elevation proceed like 1 year before this elevation. Moreover, they analyzed also the tumor content and by NGS, and also the tumor, the CT content. And as you see, this mutation called ApoBec1 was not present in the primary tumor before the neoadjuvant chemotherapy, just appears at a very low level in the tumor residual post-chemotherapy. And then you can see the elevation of concentration of this mutation during the course of the metastasis. So this is 2 examples showing that we can use also cell-free DNA for monitoring. But the main iconic example for the use of ctDNA in clinical routine is the example of non-small cell lung cancer. As you see, as you know, non-small cell lung cancer profile is necessary now to give some targeted therapy. If you have some mutation, you get some activating mutation on EGFR, it is now possible to treat patients with EGFR inhibitors. But to get this information, it's difficult sometimes to get the primary sample, so lung biopsy are usually used, but lung biopsy carry risk, blood clots, bleeding, pneumothorax, and so on. There are sometimes difficulties for patients to get a biopsy because they need to stop breathing and no coughing, so it's difficult for them because they are sick. And it's not always informative for the molecular biologists because with this biopsy you get a low number of cells and sometimes insufficient quantity DNA for analysis. So maybe cell-free DNA can be used. And here I show you 2 different clinical trials comparing cell-free DNA and cytology and biopsy for EGFR mutation analysis. And I want just underline the results here. The positive predictive value of cell-free DNA is almost 100%. Specificity also 100%, just the sensitivity is quite low. But the conclusion is it is possible to use liquid biopsy to determine the molecular profile. And we can use it, and it is with a good efficacy. This IFUM clinical trial compare the diagnostic made on tumor and the diagnostic made on cell-free DNA and the objective response of those patients. And the objective response is quite the same, a little bit more higher in patient diagnosed with cell-free DNA. So monitoring is also possible in non-small cell lung cancer. In those patients that present exon 19 deletion for the EGFR gene, those patients were followed during the disease, and in the plain line disease, the quantity of EGFR mutated exon 19 cell-free DNA. And at the beginning, all the quantity dropped, and then during the relapse, there is an elevation again of this specific mutated cell-free DNA. And if we look at the dot line, this is the mutation called T790M. This is a mutation of resistance to EGFR inhibitors. And it is here the proof of concept that this mutation is completely concordant with the presence of relapse. So, for non-small cell lung cancer patients who are mutated, they usually present resistance mutation some weeks after the initial treatment by EGFR inhibitors, but those mutations could be detected. It is now necessary to detect them because there is a third generation of EGFR inhibitors. The molecule is called Tagrisso, and it is active on T790M mutation. So this molecule, osimertinib, is approved now by FDA in February 2016, and you can prescribe this treatment in patients presenting this mutation, and you can see the waterfall plot response. In the clinical trial, the authors compare the waterfall plot for best percentage change in target lesion diameter in patients diagnosed for this mutation in tumor or in plasma, and the result is quite the same, completely equivalent. And now you have these three for how to manage those patients. It is necessary if the clinician detect a resistance to EGFR TKI to detect this mutation, and to test this mutation, you can now use cell-free DNA. If it's positive, you can skip biopsy and start third generation EGFR TKI. If it's negative, because of the low sensitivity of the test, a biopsy is required to analyze the tumor content on FFPI sample. And it was confirmed in the third part of this clinical trial in December in a publication in the New England Journal of Medicine. So this is the main news. But there are many, many research now on cell-free DNA. And I just want to show you, to finish my presentation, the use of cell-free DNA in the future with two examples, by methyl DNA and also the use in screening cancer. Circulating methyl DNA is also possible to analyze. And DNA methylation is a process that is like transformation of the DNA without change of the sequence, just the modification of one cytosine. And this DNA methylation in gene promoters is specific of organ. So it could be interesting to look at this because it is more specific of the abnormality you can get from a specific cancer. And in this publication in breast cancer, they show that a panel of genes that are highly methylated is related with survival, disease-free, and overall survival. And they compare to CTCs, and this test is more sensitive than CTCs to detect those patients. But the grail is to use the test for screening cancer and, of course, to treat patients that are not advanced in the disease. And I will just show you some data from Dennis Lowe's. This is a presentation of the last ESMO in Copenhagen. This is a proof of concept that we can screen patients with cell-free DNA. This is in nasopharyngeal cancer, and they use a specific target, Epstein-Barr virus. They enrolled more than 20,000 subjects, and in those subjects, 1.5% were positive for 2 consecutive tests, and 10% of these patients have a cancer. But the good news is that those cancers were at a very low stage, and you have in orange the percentage of a low stage patient, the patients detected by the screen test, and the blue one detected in the real life. And you see it's completely different in the repartition, the different stage, or the different stage 1 and stage 2 are curable and the other are not. This is a very optimistic and very interesting slide. So to finish in my conclusion, I want to resume with the best between CTCs and ctDNA. I think they have different kind of interests. CTCs are relevant for the metastatic process, so they can be used to detect metastasis and also to understand the invasion process. They can be used to make functional assays because those cells are viable. And also, the problem is the different technologies. It's not easy to catch them. There is also some false negative with the different system because, you know, there is also some transformation from epithelial to mesenchymal type of those cells. So sometimes it's difficult to get them. And the cost is very high. For cell-free tumor DNA, this analyte is very specific, and we get very high predictive value with different tests. The processes are robust, and you can analyze also now with multiplex tests a comprehensive analysis of tumor mutation in only one sample. You have also information about the prediction to treatment, and you can monitor patient, as I said just before, and the cost is quite low. The only problem is the dilution and contamination of this cell-free tumor DNA with normal DNA coming from different cells by apoptosis, but also from white blood cells. And the sensitivity, so, is very important. If you use a technique, you have to use a very sensitive method, like digital PCR, that is very, very sensitive, or also the MassARRAY with the ultra-seq innovation. So, thank you so much for your attention.
Welcome, Dr. Irwin.
Thank you, Alison, and thank you, Professor Lamy, for that thorough overview on the current state, the challenges and the future directions that liquid biopsy can take. It's now commonly accepted that liquid biopsy will become a clinical tool to improve our patients' health outcomes. This is because it offers the opportunity of providing real-time data on tumour biology, facilitating earlier changes to effective therapies, and delivering analysis of the global disease burden that's happening in these later stage metastatic patients. And there's the opportunity of reducing adverse events by minimising repeated biopsies, resolving the issue of undetected resistance, and overcoming that heterogeneity in the disease. So Agena Bioscience is releasing paired tumour and liquid biopsy panels right now using our new iPlex HS and ultraseq chemistries for colon and lung cancer. It's the liquid biopsy panels that I will be describing for you today and showing you evidence of some of these studies that we've been doing with our collaborators across the globe. So these are the challenges that I believe there are for clinical laboratories when analysing solid tumour tissues. When analysing a tumour tissue sample, there's frequently very limited sample quantity. Preferably, our surgeons are taking small core biopsies to minimise adverse events in our patients. And these small biopsies are put into formalin-fixed paraffin-embedded blocks. This significantly limits the amount of available DNA sample, but also gives us a highly variable sample quality. And today we're no longer looking at one marker in order to do our clinical workup. We have multiple analytes that we wish to analyze to select from an armory of effective therapies depending on the DNA changes in the tumour itself. We have the challenges of tumour heterogeneity and the frequency of those cancer cells in a background of normal cells. Invasive biopsies are rarely repeated, so they're a single point in time. And in metastatic disease, we're only sampling a single site. Now, all of these challenges will be benefited from a sensitive and accurate detection technology such as Agena's MassARRAY system. However, I can further characterize them. These first three challenges will rely on very robust, short amplicon, highly multiplex technologies that utilize a very small amount of DNA input. And I'd like to just invite you to our iPlex HS webinar, which will be happening on the 26th of April. Now, this is a webinar on our solid tissue, on our new solid tissue panels for colon and lung cancer, and we'll be having clinical data presented by Dr. Bobby Sutton from the Medical Foundation, and I'll be describing the analytical details of our new panels and our new chemistry. Specifically, the minimal amount of sample input that we're achieving with a single PCR protocol, our completely automated reporting process, and with this chemistry we're delivering a 1% limit of detection for solid tissue analysis. We're not here to talk about solid tissue analysis, we'll talk about that in a month's time. And these bottom three challenges, the heterogeneity of the tumour, the fact that it's an invasive biopsy and rarely repeated, and that metastatic disease when only sampling a single site means you're not snapshotting the entire global disease in the patients. These are the opportunities that no matter how good you are on tumour tissue won't be overcome, and they're the opportunities for liquid biopsy. So why do a liquid biopsy? Well, as I mentioned, it's a real-time analysis. Cell-free DNA is cleared with a half-life of somewhere between 15 minutes and 2 hours, and we can argue which one it is. There's a lot of conjecture about it, but whatever it is, it's short. So this really is a real-time analysis. Now, in some patients, a tissue biopsy is not possible, or that tissue biopsy is lacking sufficient tumour cells or has insufficient quality in order to do a molecular analysis. And this is an opportunity for liquid biopsy to rescue these patients. And ultimately, we'll advance through doing clinical research studies to look at the effectiveness of our therapies for those patients. So I'll now introduce to you the UltraSeq biochemistry. We do standard biallelic PCR. So this is standard short amplicon multiplexed PCR with unlabeled primers. So this isn't allele-specific PCR, it's just amplifying everything that's in the template in its relative frequency. For example, if we have a 0.1% mutant and a 99.9% wild type, we'll amplify it in its frequency that's in the original sample. Where we're doing the mutant-specific enrichment to improve sensitivity is at the single base extension stage. So we are extending with primers, annealing a primer adjacent to our mutation position and extending a single base. However, we're only including in the extension reaction our mutant-specific bases, and those mutant-specific bases have been labelled with biotin. In the next step of the process, we are then taking those primers that have been terminated on our mutant strands, and we are pulling them out with a magnetic bead that's coupled to streptavidin. So what is happening is that in our final reaction product, the only analytes that are available are those that have detected the mutant-specific sequence and extended onto the mutant-specific sequence. So that means that we're able to drive the mass spectrometer incredibly hard. For those of you who know mass spectrometry, the time for an analysis is 3 nanoseconds. That's the time for it to fly from our chip through to the detector, and it only takes electricity in order to do that. So therefore, we fire the mass spectrometer many, many times, hundreds of times, in order to detect these very rare extended oligonucleotide sequences. Now, I did mention that if, in fact, all of our template is wild type, then we will get no single base extension onto our mutant terminators, and therefore nothing will be captured by the magnetic beads, and the mass spectrum that's generated will be a flat line without these mutant-specific peaks in there. So in order to control for that, we also include in every reaction 5 capture controls, which ensure that the beads have captured the nucleic acid, the cleanup and elution steps have all gone fine. And these capture controls are illustrated in this diagram with a green dot. And we also include a process control for DNA addition, confirming that you put your DNA in the reaction, you've done your specific cycling and single base extension that allows us to differentiate a negative result. So on the right-hand side at the top, what we can see is an example of a 0.5% mutant. All 5 green capture controls are present. Our DNA control underneath the blue arrow is also present, meaning that DNA was added to the reaction, and we have a 0.5% mutation signal underneath the red arrow. In the middle, we have the wild type. So again, our green capture controls and our blue process control present, confirming that DNA was added and the process has been completed successfully, and no presence of that mutant-specific sequence underneath the red arrow. And differentially, the no template control, where no DNA was added, of course there is no mutation present, but also there is no process control indicating that no DNA has been added to the reaction. So that's the biochemistry. This is all done in multiplex format, and I'd like to introduce to you our new lung UltraSeq panel. The new lung UltraSeq panel follows the new draft recommendations that have come out from AMP-CAP and IASLC, and that is that in multi-gene panels it is recommended to analyse for BRAF mutations, extended EGFR mutations, and you'll see we've described the single base changes or single base mutations, importantly L858R, T790M, C797S, which are the first and second generation, or in fact second and third generation, I should say, EGFR TKI resistance markers. But we also thoroughly cover all of the exon 19 deletions and exon 20 insertions. I haven't listed them out. It makes for a very complex slide because we have more than 40 of the exon 19 deletions and more than 10 of the insertions in exon 20. The details of the specific mutations are on our website. Broad coverage of KRAS in codons 12, 13, 61, ERBB2 insertions that can also be detected in lung cancer, and PIK3CA mutations in codons 542, 545, and 1047. So what you can see, 4 genes, and it's 78 mutations in total for our UltraSeq lung panel, and these are all being detected from a single multiplex PCR reaction. So the point here is that with a single multiplex PCR for all of these mutations, we have minimized the amount of input down to mathematical possibilities. Now this is the point where people ask me, well, what is the input? Well, with a single multiplex PCR reaction, it's just a simple question of math. If you want to detect 1 in 1,000 mutation frequency, which is the sensitivity that we can achieve with these panels, if you want to detect 1 in 1,000 frequency, well, the reality is you need to put in at least 1,000 molecules. And in fact, to have a 95% confidence interval around sampling error, you really need to be putting somewhere between 3,000 and 6,000 molecules, which is 10 to 20 nanograms of DNA. And some of you in the audience will be saying, well, I don't have that much. And that is sometimes the reality. It will work on smaller amounts of input, but the reality is you put in 100 molecules, the best you can achieve is 1 in 100 sensitivity for mutation detection. No technology can overcome the challenges of math. I'd like to show you a study that we did earlier this year, and this is the T790M ring trial that was done in Germany by QUIP. So we were supplied with 10 blinded blood samples and 10 blinded tissue samples, and we used our UltraSeq T790M assay to do the analysis. The results itself, we detected all blood samples correctly. In the tissue samples, we detected all of the known T790M tissue samples, but with the improved sensitivity of UltraSeq, we did observe 2 samples having very low-level T790M mutations that the other participants didn't detect. Now, this is actually quite common. In tissue samples prior to EGFR TKI therapy, if you're sensitive enough, you will actually find these rare resistance mutations present. They're very rare clones until the therapy is applied, and then they're selected by the therapy. And on the right-hand side, we have some example data. At the top, so for the green dots, again, like the prior slides, they're our service assays or our capture control assays. So you can see that they are all present. And then underneath the blue arrow is our mutant signal. At the top, we have our 1% mutation frequency, and this was achieved by a titration of cell line DNA available from commercial providers. So 1% mutation at the top there, Z-score of the peak compared to the baseline noise of 36, 0.3% still very clear, 0.1% still achieving a Z-score of 15, and then down to our wild type with no specific signal underneath that arrow down the bottom there. So now I'd like to introduce our UltraSeq colon panel. Our UltraSeq colon panel is, again, following the recommended guidelines for colorectal cancer screening and having BRAF, EGFR extracellular domain mutations. Now, we've added these in because this is a liquid biopsy panel, and what we're looking for is resistance. Now, you'll notice that these are the extracellular domain mutations. They're not the EGFR mutations that we're looking at in exons 18, 19, 20, and 21 that we're looking at in lung cancer. These mutations are up on the cell surface, and they actually infer resistance to EGFR blockade therapies. They infer differing resistance, as has been published by the Bardelli group in Torino, in that some of these mutations are sensitive to cetuximab, but resistance to panitumumab, and so on and so forth. So we have included those in our panel. KRAS mutations, extended KRAS coverage, codons 12, 13, 59, 61, 117, and 146. Extended NRAS mutations in the same codons, and PIK3CA. So we've got 5 genes, 107 mutations in our colorectal cancer panel. And again, a single multiplex PCR reaction so that we minimize the DNA input and getting down to that 1 in 1,000 mutation frequency detection from that single reaction. So the first study that I'd like to explain to you is the study that we did in comparison with next generation sequencing. Many of you will have heard a lot about next generation sequencing and be wondering how our UltraSeq chemistry compares to NGS in the liquid biopsy area. So this was done with one of the leading pathology groups, molecular pathology groups in China. And we had 44 samples, circulating tumor DNA extracted from plasma that was collected at the time of diagnosis. These samples had been previously sequenced with the ion proton NGS. The circulating tumor DNA after the NGS, so this was a second aliquot that had been archived, was then processed with our UltraSeq colon panel that was screening 107 possible mutations in 5 genes. Up front, we ran our exome QC panel with just 1 microliter of each of the samples template to determine the copy number of the sample. Now, these are highly fragmented samples. As Professor Lamy explained, spectrometry does not give you a very good idea of the number of intact molecules that you can amplify even with short amplicon technologies such as we have in these panels. So we use a panel to do identity and thorough quality assessment and to determine the number of amplifiable molecules such that we can guide the amount of sample input. So the result summary is in front of you. I will quickly describe the taking note of the QC status. So all of our samples passed. We did actually exclude 5 samples based on the exome QC analysis because they didn't have a sufficient intact template remaining in the archived aliquot. And thankfully, our non-template controls are failing down the bottom there. The copy number is described in the DNA copy number column, and what you can see is it's quite broad. the number of molecules that we have available per microliter. And what we then do is, if we only have 100 molecules per microliter, ideally we want to put in 300 to 600 molecules to achieve 1 in 1,000 sensitivity. Well, 3,000 molecules would therefore be 30 microliters of that sample, but it would only be just over 1 microliter of the sample in the third line from the top that has 2,751 intact DNA molecules per microliter. In the next column is the concordant results between the NGS and the MassARRAY colon panel, so the UltraSeq colon panel. So these are the concordant results. But on the right-hand column in blue, what you will see is that we do have 14 new mutations that were detected by the UltraSeq colon panel only, and hadn't been reported by the NGS panel. So because we had consumed the second aliquot of the plasma, we didn't have any opportunity to do further orthogonal testing. So what we decided to do is to go back into the NGS data and have a look at the raw reads. So in the middle here in green, this is this additional mutation that was detected by the UltraSeq colon panel, and if it's highlighted in green, it was confirmed in the raw data of the NGS, and blue is the mutations in the samples that we still couldn't see in the raw data. Now, if we dig into the detail into the right-hand column in the NGS investigation, the top one here, KRAS G12D mutation, in 4,112 total reads, the mutation reads actually amounted to 90, and therefore they've had a mutation frequency of 2%. So that was clearly missed by the analysis pipeline in the NGS. But if we actually track further down the column to the one that's second from the bottom, we observed a PIK3CA E542K mutation. We had also observed a BRAF V600E mutation in both the NGS and the UltraSeq data, but the PIK3CA mutation was not in the original NGS data. And digging in, they had a total total coverage of 17,423, and they were able to observe mutation reads of 8, which is near enough coming close down to 0%. And you can read the detail in all of the others. What we can see is that they were in the NGS data, but that they were at very low frequency. So overall, in this 44-sample study, we excluded 5 samples because of insufficient quality. They didn't meet the quality or quantity requirements in order to go into the UltraSeq panel. And then out of the remaining 39 samples, 27 of them had concordant results between the UltraSeq and the NGS. However, we detected 14 new mutations in 12 of those 39 samples, which is a 31% increase in mutations. Rather important because these are all known, actionable, understandable mutations in colorectal cancer, in colorectal adenocarcinoma. And out of those 14 new mutations, we were able to confirm 9 of them by going back in and inspecting the NGS raw data. So now a brief study on treatment monitoring with the UltraSeq colon panel. And this study, we've teamed up with the Olivia Newton-John Cancer Research Institute in Melbourne, specifically Professors Dobrovic and Go. And in this study, it's a small heterogeneous cohort, 11 samples, where they've received preoperative chemotherapy, so standard of care chemotherapy for metastatic colorectal cancer, and specifically these patients are metastatic to the liver, so where they've received FOLFOX or FOLFIRI with or without a VEGF inhibitor. And we have 3 time points where we've taken both PET and MRI imaging and also plasma from these patients, pre-chemotherapy, after the first cycle of chemo, and then upon completion of chemo. Now, this study is actually in the process of being written up, so I'm just going to show you one of the patients in this particular cohort to show you what can be achieved. Now, the UltraSeq chemistry is semi-quantitative. We can normalize that mutation peak frequency back to our process control to get a relative frequency, and therefore determine relative drops and changes in mutation frequency in patients. So what we have here at the top, in this particular patient, patient number 6, we detected 2 mutations in the plasma, a KRAS G12D mutation and an NRAS G12A mutation. The KRAS G12D at baseline or pre-chemo is at 1.9%, after the first cycle dropping to 0.6, and after the 6th cycle dropping to 0%. The NRAS mutation, pre-chemo, 0.2%, after the first cycle, 0.1% and dropping to 0. So there's an equivalent drop in frequencies of the two mutations, just differing mutation frequencies at the starting point. And down the bottom here, we have the imaging. And this patient did achieve a complete metabolic response. What you can see on the left-hand side is our pre-chemotherapy image corresponding to the 30th of the 7th, 2014 in the plot at the top. Then in the middle there, we've got it after the first cycle. Not a substantial change in metabolic response there in the imaging. And lastly, after the final cycle of chemotherapy, which is looking much better. But what's very interesting to see is that the cell-free DNA is giving an earlier indication of response than the imaging was giving. Now we want to do some confirmation and comparison, and we chose digital PCR. So in these particular patients, we did not know what mutation they were carrying. So therefore, we couldn't do digital PCR for the entire study because we didn't have anywhere near enough DNA to screen 107 mutations in cell-free DNA. So the opportunity of the UltraSeq colon panel was, with a single reaction, we were able to screen those 107 mutations, determine which mutations were present, and then we used the archived aliquot plasma to confirm those mutations with digital PCR as an orthogonal validation method. So here what we've got is characterising the KRAS G12D mutation. Down the bottom is the digital PCR plot on the left-hand side pre-chemo, after the first cycle, and right-hand side post-chemo. And the R-squared value between the UltraSeq quantitative data and the digital PCR quantitative data is 0.9869. So confirming that these mutations that were detected by UltraSeq are present in the plasma sample, and the relative frequency that we're detecting with the UltraSeq panel is concordant with the digital PCR. So what are we, where is Agena Bioscience with our oncology panels at present? We do still have our OncoFocus panel for colorectal and lung cancer, delivering a 5% mutation frequency detection. Next month, I'll be describing our iPlex HS panels for colon and lung cancer, delivering a 1% limit of detection. And today, I have described for you our UltraSeq colon and lung panels, delivering a 0.1% limit of detection with minimized sample input. So the MassARRAY solution offers you targeted panels to identify mutations that have known significance. Genes such as BRAF, KRAS, EGFR, ERBB2, NRAS, so on and so forth, PIK3CA. We have completely minimized the sample input down to mathematical possibilities. By using a single PCR reaction, the reality is that all you need to do is put in the minimum amount of material in order to get your mutation into that reaction. We recommend starting with 10 to 20 nanograms of starting DNA for UltraSeq because that is between 3,000 and 6,000 copies, which means that you should achieve a 1 in 1,000 sensitivity, which is the capacity of the panel. We have a very fast turnaround time, DNA to results in less than 8 hours. And I showed you residual disease monitoring studies where we can detect more than 50 mutations from that single reaction down to 0.1% from a single blood draw. So thank you for your time, and happy to start taking questions along with Professor Lamy.